Okay, so everyone, my name is Harry. I'll be taking you through this video tutorial. This is section 2, the second video in this tutorial series, and we're going to look at the chemistry of the cell and the physics of neural cells, and then we'll look at how that will cover it I'll try and be as quick as I can, and we'll be very brief, and because I do want to talk to you about action potentials and synapses and neural networks. So there's a lot to cover. I'll try and be concise. Okay, so as said before, have a read of the description, leave some comments if you have any questions or any feedback to give, please do. A copy of the file, the PowerPoint, please download it and you can print it out and use it for studying. And there's more information in the description, so just below. So neuroscience is, as we saw in the last video, it's obviously very interesting, to me at least, and it can get very complicated. I just want you to get the main concepts in neuroscience. So anyway, this video is going to be about chemistry of cells, and two things: signaling within cells with informational molecules. We'll look at that. But especially, the main thing I want to talk to you about is signaling from neuron to neuron, action potentials, and that's what I want to talk to you about mainly. So we'll get to that as quickly as we can, and we'll look at some chemistry. There's not much maths in this video. Concepts in brief, and which I think you have to know for A level. Certainly at university, you will have to know these in a lot more detail. So we have different types of neurons. We have the different types of neural cells. We have glial cells. You can see the diagram here. The yellow is the neuron, yes, and you have the blue and green are glial cells, which provide support for neurons. That's what I'm going to say about glial cells. And so if you're interested in glial cells, download the link in the video description, and so download the file and go to that link. And if you want to learn more about glial cells, I don't think they're as interesting as neurons, but if you're interested, have a look at glial cells. Just Google glial cells. So what I want to talk to you about mainly is neurons. And neurons are the unit of the nervous system. They're functional units, and these are just simplified diagrams. And we have axon and dendrites. We have the cell body. That's it really. That's all you have to know. They come in all shapes and sizes, as you can see. We'll cover this more in maybe section 4 on histology and section 6 on computational science, neuroscience. So yeah, there you go. So what we're interested in is genes to organisms, and for this video we'll look at we'll look at the molecular basis of neural cells, and in the next video we'll look at organ systems. So again, as I've said in the first video, we won't cover evolution and development. I spoke about that briefly, but we'll look at the anatomy and physiology, structure and function. So for A level, I think you need to know this. We have DNA. We have the code, which is obviously the DNA molecule is three-dimensional, but the code is a one-dimensional code. And then you want to go from that code to the protein, the three-dimensional protein. That is essentially the basis of life in a way. Proteins, we have made of amino acids, the structural and functional components of cells, as I wrote there, and that's it. That's pretty much it. That's a ribbon diagram here, which I like, of an ion channel. I like ribbon diagrams. Maybe that's just me. I don't know. There you go. So if you do A-level chemistry, if you want to do biosciences at university, you need A-level chemistry, I believe. Okay, like at UCL where I went, for neuroscience and biomedical sciences, you need A-level biology and A-level chemistry. You need an A. You need to do pretty well. Chemistry is essential, and for biology, you don't have to know. You need let's see, hydrogen, carbon, nitrogen, oxygen, maybe some fluoride, phosphorus, and silicon. Chlorine ions, we talk about potassium, sodium ions. Pretty much it for, you know about carbon. That's it. I mean, we're not going to go too much in depth with the chemical elements. What we're interested in is simple molecules and how they form macromolecules. We'll talk about that. And you do have to know this. I mean, for A-level, for university, if you want to do cell biology, you have to know all the complexities of it. I mean, even this diagram here, I don't even think is that complicated. I mean, you might have to know that for A-level, I'm pretty sure. So the basis of the molecular cascades, the metabolic processes and algorithms in the cell, you have to know about this to understand how cells work. And I don't want to should I cover? I think I might cover it more if you request in the comments that I cover this in more detail. Just let me know. That's all I'm going to say about this for now. And it gets at university, if you do biochemistry, you have to know, all the pathways, all the mechanisms, all the reagents, the intermediates, what are they called? Well, anyway, you have to know, all the molecules and all the pathways involved, which is, I think, beyond my scope. So, and also beyond the scope of this video. So, the main thing I want you to get out of this is that the molecular mechanism, the molecular processes in neurons is essentially has a basis in the chemistry of molecules. Okay, as I've shown here. That's the kind of depth you have to know, is I would say for first year university, and probably for A-level as well. I mean, you know better than I do. And so there you go. So because I did chemistry A-level like I don't know what, like 10 years ago. I don't know, but you study it again in university biology, and you have to know about periodicity, bonding, kinetics, energetics, thermodynamics, molecular mechanics, the electron shells, which are actually the probability density clouds. But once the mathematics you have to study, but just the concepts of this, I want you to you need to understand this. And the basic concept is the basic concept of the biochemistry is, or chemistry, and again, I'm not a chemist, I'm no expert on this, but I know a lot about biology. You have an electron going from one place to another. Okay, it goes from here to here, from here to here. That, to me anyway, is the gist of biochemistry, or chemistry as a whole. It's obviously a bit more complicated, but as I said, I don't want to go in too much depth with this. All you need to know for this video is we have molecules, and molecules, for example, in that case, they bind to, so in that case you have glutamate binds to a receptor, and we have structural change in the receptor molecule, and for example, in that protein there, it is ion channel, for example, we have a structural change when there's binding. There's biochemical reactions, and you have a structural change in the in the functional ion channel here. These are conformational changes, and so that example, it could, for example, open the ion channel. Okay, that's the molecular basis, and again, you have to follow the electron. For molecular biology, I did second year molecular biology. You don't need to know the electron mechanisms because I specialized in systems neuroscience. So that's all you have to know for now for this video. You should know this, which is all the organelles in the cell. You have to know this for A-level. All the intricacies in the cells, dendrites and axon. Obviously, we have the cell body. We have microfilaments, microtubules, which are structural proteins, structural infrastructure of the cell, but also the functional kind of motor transport of vesicles and those sorts of things. Nucleus, nucleolus, membrane. We have mitochondria. You have to know about mitochondria. It's important. The endoplasmic reticulum, the Golgi apparatus. You should know this. And by the way, this is a kind of simplification of the complexity of the cell. This is just for A level. I mean, if you do third year molecular biology, it gets a lot more complicated than that, and you will have to know these at some point. This is one I want you to get out of this video. These are the main points. So the chemical and physical basis of cells, of neurons, leads to the structure of neurons. Okay, and also the function. And based on the molecular basis of cells and the chemistry of neurons, these that we've just described, that gives the neurons a unique functional property that no other type of cell has. It can generate and transmit action potentials. I mean, that is a fundamental principle in neuroscience. You have to know this, that the chemical and physical basis of cells underpins of neurons, underpins its ability to generate and transmit action potentials within the neuron and from one neuron to the next, which is basically transmitting, firing, neurons firing. So we'll talk about that now. And in neural networks, that becomes important because that's how information is computed in networks of neurons. Okay, so that's the main message of this video that I want you to understand. Okay, these are some Nobel Prize winners that you can that have contributed a lot to our understanding of the mechanisms. Hodgkins, Huxley, Claude, and Sackman, and Carlson, Kandel. Kandel's the guy. He co-wrote the book Principles of Neuroscience, I think. Okay, so that's that. And so there you go. So the action potential. I'll be very brief here. So it's based on potentiation. Potential, which is in electrical potential and chemical potential, or electrochemical potentials, which is the differences across the membrane. So in that case, the transmembrane voltage differences that are based on electrochemical changes. And what we have is, you can see on the right here, we have a diagram that it's not a good diagram, but you can see the plus in the top neuron. That's a neuron, that's a cell body and an axon. You have the positive inside the axon that travels through the axon, and that is the action potential. The other thing is, there's other information here. Do print out the PowerPoint and look up these, the Goldman equation, Nernst equation. You should know about these for university in the first year neuroscience. So we're going to talk about this now. So what we have is a ligand, a neurotransmitter, binds to a ligand-gated a receptor, and that opens an ion channel. There's depolarization, and that depolarization is carried through. Okay, that's the action potential, and that's based on the voltage-gated ion channels. So here, what we have is, look at that graph. You have to know that graph for A level. Okay, very simple. So you start at the resting potential, minus 70 millivolts. You get a stimulus, and then once it reaches a threshold, based on the movement of the ions, once it reaches a threshold, you get a massive depolarization in the membrane. That is when the sodium ions diffuse into the neuron, so it becomes more positive, so less negative. Then at the top point, the sodium gates close and the potassium gates open, and that's when the potassium goes out of the neuron, and that's the repolarization process. And then there's a hyperpolarization, a refractory period, during which you cannot have another action potential. You have to wait till the end, says at the end here 6, you have to wait till you get back to the resting potential. That's important, and we'll look at why now. So there you go. This is these are ion channels, open and closed. So you get a stimulus input, and then if it reaches a threshold, it fires one action potential. And then if there's still a stimulus, again another action potential. 1 1. If it's not stimulating enough, 0. Okay, it's not a fraction. It's 0 or 1. 1 1 0 0 0 0, and then again 1 1 1 with the stimulus input. You cannot have in the bottom right here. So you have a 1, but you cannot have a 2. Okay, either the action potential fires with an amplitude of 1, which is an arbitrary unit of 1, or it doesn't fire at all. You can't have 2, a value of 2. You can't have value of 0.5. Okay, it either fires with value of 1 or it doesn't. You can't have 2. You can't have 0.5. You can't have a continuous, like the black block here. It cannot be. It fires once with value of 1, or it doesn't fire at all. It either fires or it doesn't fire. And with it fires with the same amplitude, but you don't get a continuous. It doesn't stay at the peak here. See, there's a quick depolarization and repolarization, and then there's a refractory period. You cannot have, with continuous stimulus, it goes 1, pause, 1, pause, 1. It's not just 1 continuously. Okay, I hope that makes sense. You have to know this. It's important. And the other thing is, okay, so that's it for that's what I'm going to say about action potentials. But we have a transmitter that binds to the receptor. There's depolarization. The action potential travels through the axon to the synaptic bulb, the end of the axon, at which point we have vesicle release of transmitters, and again that goes through the synaptic cleft, the gap between neurons, and then again same process happens in the other neuron. Action potential fires in the other neuron if there's enough stimulus input. That you should know. The synapse is the site of drug action. So there you have it. We have the synapse, we have action potentials, and the other important thing is you have to know about long-term potentiation, which is essentially when you get repeated stimulation of the neuron, you get that particular process. And the way it works is, I don't think you need to know much about it for A level, except the fact that the calcium ions are involved. So I think what I'll do is I'll probably leave it at that. This is the review, which I think is excellent. Please download the PowerPoint and copy and paste that link. This is a 2012 paper on calcium dependent and Nmda receptor dependent LTP, and it explains it a lot better than I can, and you have to know that for university level neuroscience. For A level, not really. I mean, you know, we have neuroplasticity in the brain. There's a change in the structure, the morphology of the neuronal connections, which is based on whether or not we have repeated stimulation to neurons. I mean, Hebb said in 1940 something like neurons that fire together wire together, and we've had other work by Bliss-Lomo on this in the 1970s, looking at the molecular basis of LTP and the electrophysiological basis of LTP, especially in the hippocampus, and that involves the calcium ions as well as sodium and potassium ions, and the Nmda and AMPA receptors molecules. And I will actually, what I'll do is I'll do another video on let's see, probably in section 5 on cognitive neuroscience. I'm going to revisit that concept, or subtopic rather, because it is important in learning and memory, and I'll cover it again in, I think, section 5, cognitive neuroscience. So that's all I'm going to say for now. Look at that link. All you have to know is that basically if you have a connection between one node and another node, between A and B and B and A, that right there is what we call learning. Okay, you strengthen the connection, that is learning. And we will talk about it more in cognitive neuroscience. And we'll also talk about it more in section 6, which is computational neuroscience and neuroengineering and systems neuroscience. Yes, I think I'll leave it for these videos, because for now, I mean, that's it for now. Just, you don't have to more than this. And the other thing, because I wanted to move on to neural networks pretty quickly. So we have different types of neurons, which are excitatory or inhibitory, and you can connect them together into networks of neurons. There are not infinite, but the number of combinations of different types of neural networks is just absolutely huge. I mean, you can make, these are just 5 neurons, and you can connect them in I don't know, thousands of combinations. I mean, just add a few more neurons and a few more connections, and it gets a lot more complicated, let alone 100 billion neurons with 100 trillion connections. I mean, that is mind-blowing, I would say. And what I want you to understand is we have the molecular basis of neurons, underpins their physiology, which underpins their function and how they communicate with other neurons. Okay, I think that's pretty much it for this video. Okay, because in the next video I actually want to talk about sensory and motor systems, essentially, which are large networks of neurons. Okay, that's pretty much it. So the other thing is, recent and current neuroscience research, very interesting. Please look these up. If you're interested in this, look them up. We have developments in pharmacology. This is over the past 10-20 years, and what's happening right now, very exciting. We have stem cell regeneration of neurons, very exciting stuff. Gene therapy, which should have progressed more, but still good things in gene therapy happening. Genomics, proteomics, the Interactome project, the Human Connectome project. Please Google Human Connectome project, and there's probably YouTube videos on it. Look that up. Memories and optogenetics, that is very interesting. I did a project in my university on that, just analyzing a paper by Tanaka et al. You can actually activate and inactivate neurons by using light beams, or rather lasers, I think. And in the hippocampus, because I specialize in the hippocampus, and you can reinstate cortical memory representations from stimulating hippocampal neural cells. This is exciting. This has huge application. This is in rodents, but this has huge medical applications in humans as well. Also, just enhancing, creating new memories that are false, that are not real. And so the memories are real, but the events never took place, and you can erase memories. It's in kind of infancy of the genetics, I would say, infancy. But that's it. That's pretty much all I have to say. Look, Google it in more detail. Books. I want to show you some books. Hold on a second. Okay, just give me 1 second while I actually. Yes. Okay. Right. Okay. So we have molecular. Yes. So you need. This is a great book. It's Molecular Biology of the Cell, which is. Let me just show you quickly. There you go. By Alberts et al. It's a great book. We have. Hold on a second. We have some chemistry. This is. That was from like 10 years ago. You can see that. Actually, don't get this one. Get the Berg et al. One. We have an anatomy and physiology. That is my favorite for anatomy and physiology, which is by Tortora, I think. Yes, it is. The other one is Del Purves Neuroscience. That is probably my favorite neuroscience textbook ever. Right there on the right, and it's a great book. Please look it up. It can get pretty expensive. This is a photo from a university. I had to give them all back when I left, but I took a picture. Why not? Good memories, I guess. I don't know. But Neuroscience, it's a great book, and it covers in more depth everything I've talked about, basically. What else do we have? We have let's see. I don't know, some random books. We have some just normal brain atlases and stuff. What's this? Let's see. Basic and clinical. I don't know if you're interested. Oh, this is just a popular science book. Susan Greenfield, Stephen Rose, Making of Memory, great book. I think there's a book, Breedlove, Cognitive Neuroscience. Yes, it's in the photo here. Yeah, it is. A computational neuroscience. I'll have to find it. A book I bought. Anyway, A-level AS. I'm sorry. Is this the right one? No. Oh no, there you go. A2. Okay, that was from, like, for me, probably 8 years ago. It's in the last topic, in the final topic, neuroscience. I just realized that. Yeah. So there you go. There you go. Seminal papers. Just please download this PowerPoint in the link in the description. Download the PowerPoint and you can just Google. You have to know these. If you're at university, you're writing essays, you have to know these papers and reference them and know what the experiments that they did, because that's how we write essays in neuroscience. Young, J.C. Young, Hodgkin, Huxley, Goldman, Keynes, Armstrong, and so on. Let's see who we have. Many others. Just look these up. These are key seminal papers. You have to know. Some of them are probably, and by the way, they're kind of old school. They're like from 1930s till 1990s. But if you're on more recent ones, they're not that recent either. But look these up as well. And websites. Websites are good. You can look up Neurochemistry Journal, Neurochemical Journal. The Scientifica is useful. The NIH website, LTP, look that up. Action potential, simulations and animations. Just Google on YouTube. It's got videos on animations at the UCL websites, because I have a degree from UCL. It's very good. I hope these links work. I'm not sure. Just download the PowerPoint and copy and paste these links. Yes, and that's it. And that is pretty much it for section 2. Please read the description for more detail. Like and subscribe if you want more videos. Comments are good. If you want to give feedback or mistakes to point out that I've made, please, if you share the video with students, classmates, they might find it helpful. I don't know. That would be good. And that's pretty much it. This has been the chemistry and molecular biology of neural cells and the physics and physiology of neural cells. In brief introduction to main concepts. That's what this video was supposed to be. I hope. And at least now you know what a neuron is. That's pretty much it. You know what a neuron is. You know what it does, more or less. Action potential. And in the next video we'll look at systems networks, from vision or sensory system vision to motor functions. There'll be a lot to cover there, but I hope we can go through it, and hopefully it'll probably be more interesting than this video. Leave a comment. That's it. I guess I'll see you in the next video on neuroanatomy and neurophysiology. We'll look at the clinical aspects, which are very important. And that's it. So thanks for watching, and I'll see you in the next video.